Welcome to Population Genetics. This is an introduction to the course by me, the professor, Dr. von Webberg. This is also why I think population genetics is a critical field in biology in the early '80s, and why I think you should take this course. So, population genetics has become an essential field to many different applied parts of biology. One of those is forensics, another is breeding, a third is conservation, and a final one, likely to be most enticing to FIU biology students, is that it's central to modern medical genetics. In many ways, population genetics has become the central part of contemporary genetics. Other aspects of genetics, molecular genetics, have perhaps equally long histories, but a lot of questions we increasingly ask with population genetic approaches. And it's also a foundational field for all of evolutionary ecology. People have said that, Dobzhansky famously said that biology only makes sense in the light of evolution. And Michael Lynch, a very prominent population geneticist, said that evolution only makes sense in the light of population genetics. It's the mechanism that we use to understand all changes in populations. So this course is going to cover most of the central topics in population genetics. We'll start with about 2 weeks of background, then we're going to jump into the central principle of population genetics. It's in a way our null hypothesis, the Hardy-Weinberg equilibrium principle. And then after covering it for a week or so, we're going to spend most of the rest of the course talking about factors that cause Hardy-Weinberg equilibrium to be violated. Things such as selection, genetic drift, migration, inbreeding, linkage disequilibrium. And we'll wrap up the course spending some time on some particular examples in forensics, human medical genetics, and conservation. This course focuses on a couple different kinds of activities, emphasizing things for different learning styles that you may have among you. Every week there'll be a problem set. They're mostly going to be a handful of hand-chosen problems from the back of the Hedrick book. You'll be asked to do them, turn it in through Turnitin. There'll be a weekly discussion thread. You're expected to contribute to at least 75% of them. It's 10% of your grade. Every week there'll be a prep assignment. Often this will be writing a few definitions or discussing the importance of a topic. There's a weekly blog. The blog is tied to the term paper. I want you to blog about your experience examining the primary literature in population genetics. So the largest assignment in this course is a large 10-page term paper. At the end of the second week, you'll turn in a proposal for your term paper, what you intend to study. You'll have to include some citations in that. Then a couple weeks later, you'll turn in an outline with annotated citations for that term paper to show that you've started to examine the primary literature of the field. A couple weeks later, you'll turn in a first draft. It doesn't necessarily need to be complete, but the more complete it is, the better shape you'll be in for the rest of the semester. Each of you will peer review 3 of your colleagues' term papers, providing comments on everything from grammar to how good the question is. And then at the end of the semester, you'll turn in a final draft. That's the large assignment. You have some freedom to pick your own topic. However, it's constrained by being about human migrations or the consequences of human migration. So you can tackle anything related to human migrations. Many diseases are only prevalent in particular populations, and those diseases have a history that is due to those human migrations. So you are allowed to work on those. Many plants and animals, such as our crops or our livestock, have also been moved around by human migrations. Many environmental problems are in part due to human migrations as well. So this constraint that you write about something about human migrations and the population genetics of that should not constrain your interest very much. The weekly blog will be about your experience in generating. It will be a chance for you to tell us how you're doing with that assignment. So that's what you're going to experience in this course. There will be lectures like this posted, usually under 15 minutes, but much of what you'll do is those assignments. So I want to end this introduction by mentioning some of the areas where population genetics is increasingly useful. One is in parental testing, where we can use short tandem repeats, also called microsatellites, or single nucleotide polymorphisms, SNPs, or other marker types to figure out whether a child is from a presumed mother or, more often, a presumed father. This can also be done with mitochondrial or Y chromosome-specific loci. There have been recent cases, this one is getting a little bit older, about Anna Nicole Smith and the father of her baby. Non-paternity events are pretty common in human populations. Somewhere between 2 and 10% of children have a different biological father than their social father, with in vitro fertilization, infidelity, sperm donation, and hidden adoptions, where families arrange adoptions and don't tell anyone about it, anyone outside the family about it. These happen to be pretty common. And although they can be culturally significant, biology, population genetics, can change our understanding of these. Increasingly, we can do DNA testing beyond humans to figure out what kind of dog you have, or whether the meat in the freezer is a particular kind of horse. It can be used to track introductions, say a new invasive disease or a beetle. It can allow us to track them. So you can even go in and commercially have your dog genotype to figure out what kind of dog it is. Finally, human medical genetics. This is an increasingly very important area where in any disease with a genetic component, we use population genetics to understand the prevalence of that, and increasingly to figure out what the gene underlying the trait is. So there have been a lot of recent papers on this. This is just one from a couple of years ago. Population genetics is also critical to understanding the process in natural selection. It provides a set of very powerful tools to find the evidence in natural selection from DNA sequences alone. One place where this has been done is finding evidence for high-altitude adaptation in Tibetans. Interestingly, in the high Andes of South America, there are similar adaptations that have arisen independently, conferring high elevation adaptation in those populations. And in breeding of crops and animals, population genetics is critical. It provides a lot of tools to make better corn, better cows, better pigs, so that much of the reason that we have enough to eat in this modern world with nearly 8 billion people is due to applied population genetics, breeding, increasing yields by at least an order, if not 2 orders of magnitude, over a century ago. One of the tools to do this is something called a nested association mapping population. This is something developed for maize or corn. It's been very powerful. And finally, population genetics is critical in conservation, where inbreeding in small population sizes can exacerbate the risks of extinction in organisms of interest. And one place where this has been very useful is in the genetic recipe for the panther. So an example we'll come back to. So with this, I'd like to welcome you to population genetics and hope you enjoy the semester.